So the purpose of this lab was to really go through this process of science, right? Where do we go from, being interested in something to performing an experiment to getting results? And we started with the first part or the first step in the scientific method, which is to ask a question. Really, you find something interesting and you ask a question about what it is that you are finding interesting, right? In our case, we had two questions. The first was, do pea seeds germinate faster if they are pre-soaked before planting? And the second was, will pea seeds germinate differently if they are grown in light versus if they are grown in darkness? And from that question, you formulate a hypothesis, which is an educated guess. And it's based on information that you may have already or some research that you will do. So what you actually are doing are you're formulating two hypotheses, right? Your alternate hypothesis is if you think there is going to be an effect, right? So for our first question, do pea seeds germinate faster if pre-soaked? Based on our understanding of the steps of germination, our hypothesis, our alternative hypothesis, was that pre-soaked pea seeds will germinate faster than non-soaked seeds, right? But it's important that you also formulate your null hypothesis, which is the hypothesis if there is no effect. And that's exactly what we have spelled out there, right? Our null hypothesis is that there will be no difference in germination speed. Why that null hypothesis is so important is when we are analyzing results, which we're going to get to in a second, we are actually analyzing those results based on that null hypothesis, right? And our results are going to determine whether we accept that null hypothesis, and there really was no difference, or we reject it, right? And if we reject it, it means that there is some difference. Now, it may be what we're suggesting, right? That pre-soaked may have done it, but it may be some other explanation. But we are always comparing our results against that null hypothesis, which is why it's so important that we formulate it in this step. Right? So for our first question, our first experiment, that's what we're looking at. Our second experiment, we were interested in whether there was a difference in growth between seeds germinated in light and seeds germinated in darkness. Right? And for our alternative hypothesis, we didn't have as much information, but we knew that light is important for plant growth. So we are going to say there is some sort of difference. And the null, again, was that there was no difference in growth. Now for our experiment, we went over that. We soaked some of our seeds. We grew them under light. Some of them we grew them in darkness, right? We had multiple treatments. And each day we measured the radical and the stem length, and we collected our results. And now it's time to move on to step 4 here, and we're going to analyze our results. So how do we do that? Well, we use a branch of mathematics called statistics. And really, statistics is just this field of math that deals with data. And that's what we collected, right? We have data on the length of our roots and our shoots and each time of the day and when they started germinating, when we first saw them. So now we need a way to analyze this data, and we use statistics. So I'm not going to go through. Statistics is a large field of math. It can get very complicated. And what I really want to do here is just kind of go through an introduction to stats and how we use it in a scientific experiment. And I'm just going to kind of gloss over this. I'm not going to go too deeply into these topics, but I just want to give you enough where you have, if you see this in the future, you will have a bit of an understanding. But I also encourage you guys, if you guys are interested in science, that you should take a stats class at some point because it is the way that we analyze our data in our experiments. So we have a bunch of data that we collected. And one of the first things that we often want to do is describe our data. So we use a bunch of tools in stats that we call descriptive statistics, right? And these descriptive statistics, these tools that we're going to use, are really to let us know what our data looks like, to get insights into our data. And while descriptive statistics may sound complicated, it's really not. It's stuff that you have used throughout your life already, or have seen throughout your life already. And perhaps the most common descriptive statistic that we use is the mean or average, right? We have collected a whole bunch of data points of a sample. For our case, we're going to say we have a whole bunch of data points for the end growth, the end length of our stems, right? So there are 9 of you plus me doing this experiment. We had 3 plants growing in each treatment, so we should have 30 plants, a sample of 30 plants. Right, so we have this sample of 30 plants. And at the end of our 14 days, we will have a length measurement for each one of these plants, for each treatment that we did. Right, what we may want to do in the beginning is, what is the average for all these plants, right? We know that not all of these plants are going to be the same size. There's some inherent variation in the plants themselves, maybe where they were grown. We probably all did our experiments slightly differently. That's going to lead to variation. That could lead to different end lengths of our plants. So the first thing that we want to often do is calculate what is the average. And this is an important step, and it gives from this we're going to kind of build off from that. Right, so you calculate the average of your sample for, you all probably know how to do the average, right? Add up all of your things, divide by how many there were. So that's the first start. So average tells us one thing. But another important descriptive statistic is variance. Right, variance is going to tell us how spread out your results were. So I'll give you an example. Right, down here, say we just have 4 plants, right, and we have, this one is 5, 5, 6, and 4. These are the end stem lengths, right? So at the end result, if we're going to calculate an average, right, we add all these up, 10, 20, divide by how many there are, 4, 4, and we get an average stem length of 5. We'll say this is just centimeters, right? We can also have something like this. 4 plants, 10, 1, 6, and 3. Right, and we add all these up, 10, 11, 17, 18, 19, 20. Divide by how many we have? 4, and again, we have an average of 5 centimeters. Right, so we have 2 different samples here, where in this treatment, we have an average of 5 centimeters, and in this treatment, we have an average of 5 centimeters. But you can see, if you look at the individual plants, there's a big variation between them, right? Here, all of our plants tended to be around the average, whereas here, they're much more spread out. So that measure of spread that we see here is our variance, right? It's a measure of spread from the mean. And that's important because that is because just as your mean will tell you something, this variance is going to give us information as well, excuse me. So oftentimes, we can view our data looking like this bell-shaped curve, right? And this would be like a histogram of our data. So right here in the middle is going to be our mean, our average, right? So if we're looking at a large population, if we're looking at, say we are looking at the population or the height of students at the Cumberland campus, right? Generally, most of the students are going to average be around this area, right? They're going to be whatever the mean is. But then you may have some particularly short students, right, that fall over here, and are particularly tall students that fall over here. And when we're looking at the y, this is just like the number of students, right? So most of our students are going to fall close to the average. Some are going to be particularly tall, some are going to be particularly short, right? And this entire spread is the variance. Now, we often calculate variance as standard deviation, and we're going to see that when we get to the next part of this, right? And we can get more in depth on all this stuff, but I don't really want to go there. I want to try and keep it mostly visual. So, what are we doing when we are testing our hypotheses? Right? We are essentially asking the question, if we have, for our case, we have two samples. Right? Let me go to a new thing. And we'll start with the one hypothesis. So we'll say our null is that there was no difference in end height of stems no difference in end height of stems in plants germinated in light and dark, and their alternative that there essentially was a difference. All right, so this is where we are looking at the effects of growing our seeds or germinating our seeds in light and dark. And how we're going to measure it is by looking at the end height of our stems. And if we see a difference in the end height of our stems, that's going to suggest that there is some effect of light and darkness on our growth of our plants when we germinate them. So we set up our experiment and we will have two samples, right? We will have those grown in the light and those grown in the dark. And at the end, we will have 30 replicates or 30 plants, and we'll have values for all those 30 plants. I'm just going to put random values in, yada yada. 18, 5, 7, 20, 23, 6, 17, 18, 4, 30. Right, so we'll have two data sets, and we have one data set of our plants grown in light, other data set of our plants grown in the dark. And we want to compare these data sets because we are interested in seeing, is there a difference between them? Right, so we have, really what we're looking at is we will have, if we calculated our mean and variance over here, we would have our little thing, our little bell-shaped curve that looks like that, and we'll have a separate one over here for our dark. And we're trying to figure out, are there really two separate curves here? Or would this data set make more sense as being actually one data set? That's what we're looking at. That's what we're trying to figure out. Are there two distinct data sets that we see here? Or is it more likely that it's just one big data set that incorporates all of these values? So this two distinct bell-shaped curves is indicative of our alternative hypothesis, whereas this single curve is our null hypothesis. Right, so to evaluate this, we are going to use what's called a t-test. And a t-test is a statistical test that we use to evaluate differences in two populations. Right, so And really what we are asking is, given the mean and the variance in our two populations, are they significantly different to actually be two separate populations, or are they more likely to just be one large population that has that includes all of these values? So we perform this test, and we're not going to go over how to perform the test, we're just going to do it in Excel. But we will perform this test, and at the end result, we will get a test value, and then we will get a p-value. And that p-value is really important because this p-value is essentially, when we're talking about hypothesis testing, it's essentially the probability of getting this result if the null hypothesis was true. So let me write that out. Probability of getting this result if the null hypothesis was true. Right, so we are going to compare these two treatments and the values between them, and we're going to look at the mean and variance. And it's going to give us, using this t-test, and the end result, it's going to give us a p-value. Maybe something like this, 0.05. And this p-value tells us that there is a 0.05, if this is our result, it tells us there's a 0.05 probability that given the data that we collected, the null hypothesis is true, that there is no difference between these populations. Right? And this 0.05 is really important because this is the cutoff number that scientists have agreed upon as being statistically significant. So if you run a statistical test and you get a p-value of 0.5 or lower, it's considered statistically significant, and that we are seeing a true effect or some sort of true effect, and that you should reject the null hypothesis, that there isn't any difference. Right? So essentially, again, to reiterate, when this p-value is telling us the probability that the null hypothesis is true, and if the probability is 0.5 or lower, we reject that null hypothesis, and that suggests that there is a difference in our treatments. In our case, it suggests that there is a difference in the growth of our plants if they are grown in light versus if they are grown in dark. All right, so hopefully that makes sense. We are now, I'm going to switch over and I'm going to pull up our data set that we collected, and I'm going to show you how to run this t-test on Excel and calculate our p-values. And then from that, we can kind of get some insights into our data. Okay.